My name is Edgar Nunes, and we're going to start off by the G protein pathway. First off, we have the G protein linked receptor right here, which is the blue figure, and the ligand, which is the red figure. The ligand is a chemical that can't make its way through the cell membrane, so it docks itself to the top portion of the G protein receptor. Now, the G protein receptor has two portions, the top portion and the bottom portion. The bottom portion is made up of subunits. When the ligand attaches itself to the top of the G protein receptor, it changes the shape. When it does that, a subunit is released, also known as an alpha subunit, or it can just be a G protein. The G protein travels on to the adentylyl cyclase right here and attaches to it. Before the G protein was even there, the adentylyl cyclase was inactive. Once the adentylyl cyclase becomes active with the G protein or the alpha subunit, it makes CAMP, shown right here. It converts the ATP to cyclic AMP. The ATP drops off two phosphates, creating AMP, but a cyclic portion is still left, creating the CAMP. Now, the CAMP goes on to target the protein kinase, which is this right here. The CAMP binds on to the regulatory subunits right here, and a protein kinase is made up of two portions, which is the two catalytic subunits and the two regulatory subunits. Now, what the CAMP do, they bind onto the regulatory subunits, releasing them from the catalytic ones. Releasing the catalytic portions. Now, when the catalytic portions are released, we have a cascade energy. This is where ATP comes to the catalytic portions and phosphorylates them. Once they're phosphorylated, which is ATP phosphorylating them. Once they're phosphorylated, they become activated. Once they're activated, now they can go on into act on enzymes within the cell. Now, what they do, they go on to the endoplasmic reticulum, which is this. Here come the activated catalytic portions and go to phosphorylase, which is this right here. Phosphorylase is inactive at the moment, so what the catalytic portions that are activated come and they go to the phosphorylase and activate it by dropping off two phosphates. So now the phosphorylase becomes activated and goes through the endoplasmic reticulum, releasing glucose from glycogen, which is, it releases them once it goes through it. And they all get released and go to wherever they need to go. Once we don't have the ligand anymore attach itself to the receptor and unattaches itself, the whole pathway shuts down. Failing to mention, the green triangles are representing the secondary messengers that are also known as chymodulin and IP3. These are mainly involved in helping to create and control muscle contractions within the body. Up next, we have the tyrosine kinase pathway. The red figures right here are representing the receptor tyrosine kinase proteins, and the blue bars right here are representing the ligand. The receptors at the moment are monomers and are inactive. Now, what the ligand does, it attaches itself to the top of the receptors, making them into one whole unit, as shown right here in the process, making the one whole unit, also known as a dimer, in a process called dimerization. Even though it becomes a dimer, it's not activated fully because it's unphosphorylated. But the process is achieved by the kinase activity of the receptors. Now what happens is that GATP goes on to become GADP because it transfers the phosphates from ADP to tyrosine amino acids present on the receptors, and the phosphates go on to attach themselves to each tyrosine part of the receptor. Now, the dimer is activated and is phosphorylated. After phosphorylation occurs on the receptors with phosphates attaching to each tyrosine and is activated and phosphorylated, it can be recognized by proteins inside of the cell. Any such protein binds to a specific phosphorylated tyrosine, undergoing a resulting structural change that activates the bound protein. So what happens is that relay proteins, which is this right here, go on to bind to each phosphate and tyrosine bond. So what it does, it goes on to bond to each one, as so. And before they were binding to the receptors, they were inactive. But since the receptors were activated and phosphorylated through phosphorylation, they recognize the receptors and go to it, triggering the transduction pathway and creating a cellular response. Next, we have the ion channel receptors that are found at synapses of neurons, but this is also known as ligand-gated ion channels. First off, we have the ion channel receptor protein, which is the purple figure right here, that contains kind of like a gate, and it's like a gate area. Well, it will open and close depending if a ligand attaches to it. Outside of here is the extracellular fluid that contains all the ions, which is all the green, dark green, and lime green circles. So what happens is that the ligand, which is represented by the red figure right here, comes and attaches itself to the ion channel receptor protein, opening up the gate for ions to come through. And this continues to happen until equilibrium is achieved, because before the flow of the ions happened, there was a high concentration of ions up here compared to the bottom part, which was a low concentration. So now ions travel, but the thing with this is that only the gate only allows some ions to travel and can be kind of known as let's say selectively permeable because of that. So the ions come over here, and as you can tell that the lime green ones aren't coming in, only the dark green ones, proving my point of selectively permeable type of deal. To add on, the inflow of ions from a high concentration gradient at the extracellular fluid to the low concentration gradient at the inside membrane is provoking a cellular response. So the ions continue to flow through the open gate, but what happens now is that the ligand, which is known as a signaling molecule, detaches itself from the ion channel receptor, closing the gate, stopping the flow, concluding the equilibrium. Finally, we have the intracellular receptors right here. The orange figure here is representing the receptor protein, and the red figure here is representing the ligand. So what the ligand does, it enters the cytoplasm and moves on to find the receptor protein. So what it does is going to bind to it, as shown, and activate the receptor protein. The receptor then goes on to be a signaling molecule that acts as a transcription factor, as shown here. Once it becomes activated and turns into a signaling factor, it goes on to travel throughout the cell to go to the nucleus, as shown. It's going to travel to go to the nucleus. Once it gets to the nucleus, it's going to go on and attach itself to the DNA. The attachment of the protein receptor protein results in the promotion or suppression of the transcription of the genes to the molecule. This is the response which most common results in a new protein. So what then happens is that a degrading enzyme comes along, and binds the receptor protein to destroy it